Hi everyone.
Hi, Lilith.
I'm gonna make you co-host.
Okay.
Okay.
Mm. Okay.
Morning.
Morning.
Good morning.
Good morning.
Hi everyone. Let's wait a couple of minutes and then we can start.
Okay.
Muge, you can also try.
Share.
No, like get ready to share soon the link.
Mhm. Oh, the link. Okay.
Yeah, link to the paper.
I see.
Hi.
Mhm.
So
So I shared
yeah,
the.
maybe we can start. So
I shared the link.
Okay. One second. Yeah, good. Thank you. So Muge is today is Neuroccino 9:30, and usually we present here interesting papers. We discuss them, and yeah, everyone can do that. So this is pure volunteer work, and yeah, today Muge will present an interesting paper.
Mhm.
She shared the link in the chat, and whenever you're ready, you can share your screen and tell us about this paper.
Okay. Mm. Okay. Um, okay. So hi everyone. So today I'm going to present this paper, Clinical Severity in Parkinson's Disease is Determined by Decline in Cortical Compensation. So this is a quite a recent paper that came out at in September 2023. And then it's done like here in the Netherlands in the Donders Institute. So a group of some neurologists published this paper. So they basically, in this paper, the basic main hypothesis is that there are interindividual differences in the clinical severity of Parkinson's disease, and these interindividual differences are not due to the dysfunction in basal ganglia, but it is due to the compensatory cortical mechanisms. So each Parkinson patient has a different level of compensation, cortical compensation to compensate for the dysfunction of basal ganglia. So the interindividual differences is based on this compensatory mechanism. So the data comes from a huge data set that was collected at the Radboud University. So they analyzed data from a big project with 353 patients. And think about these patients is they are usually like early disease patients. So they have, it's been under 5 years since their symptoms started. And they also include 60 healthy controls. So in this paper, they compare Parkinson's disease patients with controls. So they have an analysis for that. And then next they compare different clinical subtypes of Parkinson's patients. And then they also look at how brain activation is associated with some severity, symptom severity. So we will talk about these. So like different symptoms, like bradykinesia and cognitive performance scores, how these are associated with brain activation. And then at the end, they show that it's the parietal and premotor regions of the cortex that compensate for the basal ganglia dysfunction. And this is like this compensation level is different for different subtypes of Parkinson patients. So there is this variability in Parkinson's symptoms, they say, can be explained by this compensation neural activity increase in parietal and premotor regions. So, okay, how did I do that? Okay. So let's start with a little bit background. One of the most common symptoms in Parkinson's disease is bradykinesia. So this is like slow motions. Okay, so these patients have difficulty initiating motions, and then they have difficulty choosing between and executing different motor actions, motor plans. And so they focus on this bradykinesia symptom in this paper. And then so they had this data comes from this personalized Parkinson project in Radboud. So it's 353 participants, and then they do an action selection task during functional MRI. So I will tell more about this action selection task. And so why do they hypothesize that compensation underlies interindividual differences in symptom severity? So they in their background they give various reasons. The first reason is that so in Parkinson's disease dopamine levels reduced in the striatum, but these dopamine levels reduce gradually. It's not like it's gone in one day. It's reduced gradually over several years before the patients start to show any symptoms. So when these dopamine levels are declining, so this basal ganglia dysfunction should be already compensated by some other mechanism. So that's why they suspect that there's other compensatory mechanisms. And the second reason is that even after the onset of disease, onset of symptoms, the motor symptoms still, decline. So even after there is no dopamine left in the striatum, the motor symptoms still, decline. So that's why it's not just the dopamine depletion in the striatum, they say. I mean, there must be other mechanisms involved. And the third reason is there are other studies that show that there is no correlation, no association between motor symptom severity and striatal dopamine levels. So that's why they suspect that there's a cerebral compensation. There some region in the cortex needs to be upregulated all that time to compensate for the lack of dopamine for the dysfunction of basal ganglia. And so they also, in this paper, they also look at the differences between different clinical subtypes of Parkinson's disease. So they had a previous paper that I just, quickly checked. They have a way of classifying these different subtypes. So they call these, so this subtyping is based on, different pathology. So they have this focal versus diffuse propagation of α-synuclein protein. So this α-synuclein protein is mutated in Parkinson's disease, and this pathology, this mutation, can start from the cortex, and then in that case, the pathology is more local. It's limited in a region in the cortex, and the symptoms are more mild. But if this pathology starts in the peripheral nervous system, they say that this pathology is more diffuse, and then you can see symptoms in various different cognitive domains, like, I don't know, like memory or like semantic fluency. Say they have the, more domains are affected if this is a diffuse type. So they have these, focal versus diffuse type, and then they classify patients into these different clinical subtypes based on this, pathology and some, symptoms in different clinical domains, cognitive domains. So we will also look into that in more detail. And so as a, for the focus of this cortical compensation, they focus on the parieto-premotor regions, because they say that there are, several previous papers and meta-analysis of functional fMRI data that shows that in the Parkinson's disease, people have more, exaggerated activation in this region compared to healthy controls. So that's why they think that this cortical compensation must be coming from that part of the brain. Okay. Okay, so they first compared these clinical subtypes that they have. So they have 3 clinical subtypes: a mild motor-predominant, an intermediate, and a diffuse-malignant clinical subtypes. And as I said, they, define these clinical subtypes based on, different motor symptoms, like different cognitive performance, sleep behavior, and, differences in their autonomic functions. And then they also quantify the relationship between these interindividual variability in brain activity, and they have this clinical ratings for bradykinesia severity and cognitive performance. Okay, so a little bit about the methods. So there, the data came from 367 patients and 60 healthy controls. And then they did a action selection task during fMRI. So, and then let's first look at this task a little bit. So here in this figure, they show the task. So this is an action selection task. Participants were shown these, circles, and then in each trial, they that one of these circles are filled, or 2 of these circles are filled, or 3 of these circles are filled. So 1, 2, or 3 choice, and then the patients are holding holding a response button. So, for example, they need to respond by pressing the button of the filled circle, right? So here is only one circle is filled, so they press that corresponding like second button here. But if there are like two choices, like two circles are filled, or like three choices, three circles are filled, they are instructed to press only one of the filled circles. So either this one, or in this case, either this one, or this one. So they can't press all three or all two. So they need to choose there. So the more buttons, like the more filled circles you have, then you have more choices. So the demand, action selection demand increases, and it's like put an load, like increases the cognitive load. And there are also catch trials. In this catch trial, for example, the second circle is filled, but in that case, they need to avoid, like they need to withhold pressing anything. So this was the task. And then they looked at for the behavioral performance, they looked at like the response time. So they need to accurately press the filled button, and then they need to do it fast. So they looked at the response times. They looked at errors and misses. Okay, so they were also looking at like response variability, like measures like response variability or response switching, but they didn't put a huge emphasis on these behavioral responses in this paper. So basically, response variabilities, you need to use all 4 buttons, right? You shouldn't only stick to 2 buttons for your response. And like response switching is if there are like 2 same trials, you should use different button presses to respond to those 2 same trials. But they don't, I mean, this is not they don't put a huge emphasis on these. So the main things is response times, errors, and misses. And okay, so here they talk more about clinical subtyping. So I already mentioned that they have these 3 clinical subtypes, like the diffuse-malignant, intermediate, and mild motor-predominant. So the most important thing here is to know that this diffuse-malignant subtype shows the most severe motor symptoms, and more impairment in more clinical domains. And this type of disease progression is faster compared to the mild motor predominant subtype. Okay, so and then as I mentioned, they look at bradykinesia severity, and then they look at like cognitive performance differences, and then they calculate some scores for bradykinesia severity, for example. They have all these, like if you look at this table, they have all these different measurements, tests for Parkinson's patients. For example, for bradykinesia severity, they use this MDS UPDRS-3 test. So it's like the clinician basically do some kind of tasks like finger tapping, walking, and then they evaluate their bradykinesia severity. And for cognitive performance, they have a composite score. So basically, they test different aspects like visual spatial perception, executive function, semantic fluency, processing speed, working memory, episodic memory. So and then they combine all these scores to come up with a composite score for the cognitive performance. And so the image acquisition was done by like a 3 Tesla MRI machine, and then they focus basically on a group level. They focus on basically 2 contrasts. So they look at the motor-related activity. So to calculate the motor-related activity, they do a conjunction analysis of 1, 2, and 3 button press choices with respect to baseline, and then they next focus on the selection related activities. So how the brain activity difference when you have two choice to choose from, or you have one choice to choose from, or you have three choice versus one choice to choose from. Okay, so, oops, sorry. Okay. Okay, so we can maybe start with looking at this figure. So this figure summarizes the differences between controls and Parkinson's disease patients. So we already went over the task. So if you look here, so in this figure, they show that the response time is always higher for the Parkinson's disease patients compared to the control. So this is when they do the button press. And also when they have different number of choices, 1, 2, 3, the response time also increases significantly for both Parkinson's disease patients and for the controls. And it's for the errors too, like number of errors in this behavioral task, they show that the patients have higher error rates compared to controls. And then they compare the activity in the basal ganglia, specifically in the left putamen activity between controls and Parkinson's patients. And then they show that Parkinson's patients have reduced activation in the left putamen. And so yeah, we see that here too. So the blue color shows that the healthy controls have more activation in the putamen compared to the Parkinson's patients, and also in the parts of the cerebellum. Okay, so this was the influence of disease status. So comparing Parkinson's patients versus controls. Next, they look at the differences between different clinical subtypes. So they show it. They show it in this figure. So here we see the behavioral response differences on the top panel. So they have these 3 clinical subtypes: the mild motor-predominant, intermediate, and diffuse-malignant. And they show that the response times were, longer for these intermediate compared to the mild form of Parkinson's. Interestingly, I thought there is no difference between the diffuse malignant and the mild form. Yeah, I don't know. They didn't, I think, or I missed that part. They didn't explain why there's no significant difference between these two. But yeah, if we look at the number of errors, then the mild type participants make less errors compared to these diffuse malignant type participants. I think this part, so this premotor cortex activity, they compare between clinical subtypes. I think this part was important in this paper. They show that these premotor cortex activity was much larger for the mild subtype compared to the intermediate or compared to the diffuse malignant subtype. So these mild form patients have more exaggerated, larger activation in the premotor region. And also in this, parietal region. So this is the, I think, main thing that they want to show in this paper, that in the mild form of Parkinson's disease, there is more compensation for the basal ganglia dysfunction in these premotor and parietal regions. So then next they compare Okay, so they then so they looked at the differences between healthy controls and Parkinson's patients, and then they looked at the differences between different clinical subtypes of Parkinson's patients. And next, they compare subtypes and controls. So here, for example, they look at the brain activity, like neural activity in the inferior parietal regions between controls and these, mild type patients. And then they show that compared to the controls, mild type patients have more like exaggerated activity in this, inferior parietal regions. And they also in the so they also compare controls and diffuse-malignant type, but this time they show the opposites. The controls have more activation compared to the diffuse-malignant type in the, like, for example, the left premotor regions. So this figure is important because it's showing that this compensation, this exaggerated activity, neural activity, compared to healthy controls, is only observed in this mild subtype of the Parkinson patients, not in the more severe malignant type of Parkinson's. So they don't have that compensation anymore, I guess. Okay, and in how am I doing in terms of the time? Okay, I have some more minutes, I think. And then next.
Yeah, but would be nice to like have some also time for questions. So I have a question also. I want to discuss it. But yeah, sorry for interrupting.
Oh, okay, no problem. So then I think there are like 2 more figures. Yeah. So I'll quickly go over the figures then, and now we can go over the questions and discussion. Okay, so and then here they look at how these different behavioral measures change with disease severity, so bradykinesia severity, and also like cognitive performance. So here we see that for more like severe bradykinesia, the response times increase, and the cognitive performance, as the cognitive performance of the patient declines, the response times also increase. Here we see that. And then they look at the, again, activation. They correlate the brain activation with bradykinesia severity. And then they also, with cognitive performance, the important thing here is to look at these, basal ganglia responses. So this is the putamen, responses from the putamen. They don't change. I mean, they are not correlated with symptom severity. They're not correlated with bradykinesia severity or cognitive performance. But these cortical regions, like superior parietal lobule and premotor cortex activation, actually correlates with the bradykinesia severity and cognitive performance. And so lastly, they have this model in their paper. So to summarize their hypothesis, they're saying that, so this is the symptom onset. So before the symptom onset, basically, nobody, there is no, there is no problem with the motor performance, right? The motor performance is functioning, it's well functioning. But the basal ganglia function is already has been declining due to dopamine depletion, right? And then they say that before symptom onset, these, the compensation in the cortical regions, these parietal and premotor regions, already increases to compensate for this basal ganglia dysfunction. So that's why for a while nothing's noticed. But then after a while, with the symptom onset, the basal ganglia dysfunction is so going declining so much so that this, even with this compensation, like this exaggerated activation of the cortical regions, it cannot still compensate enough. So we start seeing these, decline in the motor performance. So that's, that's basically what they're testing for. And so this is just a nice model to summarize their hypothesis. So I guess, so in conclusion, they also look, okay, so where is, I'm sorry. So in conclusion, they say that we shouldn't, the neurologists shouldn't always target to treat basal ganglia dysfunction. This could be an alternate, approach to actually treat these, brain regions that compensate for the basal ganglia dysfunction. So maybe we should, in our treatments, we should focus on upregulating those regions that compensate for the basal ganglia dysfunction. So I guess maybe I'm it's almost okay. We can maybe do the discussion and question part together.
Thank you, Muge. It's a really great paper with packed a lot of information in it. Are there questions? Okay, Eduardo, go ahead.
Yes.
Hi, thank you, Muge. Very interesting and dense paper. I have a few questions. One is if they actually analyze or tell somewhere if possibly the premotor compensation or premotor exaggerated activity can be explained as for example, as a function of the onset of the disease, maybe.
Function of the onset of the disease?
Yeah, maybe like patients who have been diagnosed earlier or later in time have this pattern of, so that basically it's the onset of the malady or the disease that can explain this pattern of activity rather than the subtype.
I think they cited in the introduction, they cited some papers that's based on the the gene mutations. They have these patients who are in a high risk group for developing Parkinson's disease, and then I think in those high risk groups, even before they were diagnosed, just based on the gene mutation patterns, they see some kind of increased activation in these cortical regions, I think. So so I think that's one of the ideas that's been shown in other papers.
Okay, interesting. And my second question is more kind of theoretical. So what do they mean exactly by compensation? Because if I mean the motor circuitry of the basal ganglia and the motor cortex has been very well established, more or less, so we know how it works, and hypothesizing that there is a pareto-prefrontal network that can somehow compensate for that function, it's a very strong hypothesis. So how do they support this view if they try to do?
They basically say that the activation in these motor regions are upregulated. So it's they have these it's not just regular upregulation. It needs to be, they have these criteria, so it needs to be increased. The activation needs to be increased or upregulated when there is a high demand. So they claim that they show this, like when they by parametrically changing their this action selection demands by adding more choices. So they show that it's not just patients have more activation there. It's just they engage these regions when there is more demand, more cognitive load on an action selection. And then they, I think, also looked at voxel-based morphometry. So they looked into these regions that are functionally upregulated. They show increased activation during action selection, and only in these regions they compared the gray matter between patients and controls, and then they don't show any differences in these regions. There is no gray matter volume differences in these regions. So they say that this mechanism probably is due to some functional neuroplasticity, but they don't specifically talk about which regions, which networks are included. They say that if this is a subcortical cortical network, and probably some basal ganglia regions that are not motor related take over, and then they are involved in this upregulation of cortical activation, but they don't have a specific network model of
Okay, thank you. Yeah, I was curious about that. Also given the paper
Sure.
Lily discussed a few Neurocinos ago.
Mm-hmm.
Thank you very much.
Yeah.
Okay, next one is Elvel.
Yeah. Thanks for the presentation. Just to discuss about what you said about the voxel-based morphometry differences. So it's good they did structure to check if there was any bias between structure and functions. And I was wondering if, because I checked just the supplementary materials where they show their voxel-based morphometry results, and I was wondering if in their discussion they discussed about because it's true that they didn't have differences for the parieto-premotor areas, but they do have differences for the basal ganglia, and I don't know if they discuss if that could explain why they don't have any relationship between symptoms and function in the basal ganglia, because as they have atrophy in the basal ganglia, if did they take into account these differences for saying that doesn't affect the relationship between symptoms and activity in the basal ganglia?
Yeah, I don't remember if they are discussing these differences based on voxel-based morphometry, but yeah, they found so they looked at different regions for the voxel-based morphometry. They found differences in different regions, like visual regions, and I don't remember exactly, but
It's
yeah,
in
but
the it's in
yeah,
the
it's in the supplement.
supplementary materials. So, but
Yeah, I think yeah.
and
Okay.
they got something in the temporal occipital regions, but also in the basal ganglia, all the substructures. So
Yeah. Okay. Okay, sorry. I couldn't find it now. Yeah, they didn't find these differences in the compensating regions, but some other regions, and then they basically speculate that different subtypes have different pathologies, so diffuse versus local pathologies. That could also underline why we don't see any association with symptom severity with basal ganglia dysfunction, but maybe this this severity is coming from maybe structural differences in different clinical subtypes, because each clinical subtype have different kind of pathology.
okay.
But yeah, I didn't see any further discussion on that. Yeah.
Thanks.
Yep.
Okay, can I have a quick question? So,
sure.
So I'm a bit confused because, as I understand, so there are three types. The one that has severe motor symptoms, medium, and the light, right? And then we have patients that have higher upregulation of that cortical network. lower and the middle one, I guess. And then there we have this molecular kind of explanation of that. We have α-synuclein that can be focally propagated or diffuse. So I don't, how are all these three levels are connected in this paper, or they are just like, I don't see why they're mentioning the α-synuclein.
Yeah, I think it's it's something from their previous work, like these so there are interindividual differences in Parkinson's disease severity, and then they say that these interindividual differences are usually based on the progression of the disease pathology. So in every patient, this disease, sometimes the pathology starts from the cortex, sometimes it starts from the periphery, and then they lead to different, severity, symptom severity. For example, the ones that start from the cortex are local. The ones that start from the periphery is more diffused, and then they show deficits, cognitive deficits in multiple cognitive domains. So, and then these kind of disease progress faster, right? And then this diffuse versus local pathology shows correlation, like is used to define these three clinical subtypes that they use. So if you can just ignore the diffuse versus local thing, pathology, α-synuclein pathology part, and then if you can just focus on the the three subtypes, it's, I think, enough to interpret this paper.
So did I understand correctly that a person that has, for example, diffuse propagation of α-synuclein, but has this like very significant upregulation of that network, can show in the end low motor severity because of that upregulation, even though he has diffused the α-synuclein propagation?
No, so here they show the opposite. The diffuse version, diffuse-malignant version, don't have that upregulation.
Okay, I get it.
Only the mild motor-predominant
version
shows that cortical compensation, that upregulation in the cortex.
Okay, yeah.
So yeah, I think the terminology makes it a bit more complicated, but it's just that there are three subtypes. One is more mild, one is more like malignant. The mild type shows the enhancement in upregulation.
Okay. So I think we're over time. Yeah, but it was
original for me.
It was very.
I will. Yeah.
No, it was really great, really, and the paper interesting, discussion, and a lot of questions. So, yeah, thank you again, Muge, and everyone for joining and for discussion. Have an exciting week in science, and see you next week.
Yeah.